Hi, guys.
What's up? Okay, you have a lab to complete, and this lab is kind of like a big kid lab. It's got a lot going on.
Oh, it's so much fun.
But it's really important you pay attention to the procedure, too, when you're doing it, because it's easy to mess this one up.
Okay. I mean, this is like a little chemistry going on here, and there's some big kid science going on, so it's kind of exciting. So this is the photosynthesis respiration lab. You had to wait until you learned about respiration in order to do this, but you'll witness both photosynthesis and respiration.
So you need to gather your materials first. Graduated cylinders should be out. Look on the drying rack if you don't see any out and about. The bromthymal, that blue dye.
But not methylene blue, bromthymal blue. They're two different, got names.
Out as well. That does not have to be refrigerated, so it'll just be out on the corner or on the counter.
You'll need foil, flasks, a light, Sharpie, straw, and then Elydia. So on your notebook page, write the title, Photosynthesis and Respiration Lab, and write your problem statements here. There's kind of two there going on.
Pause this if you need to, because we're going to move on.
Oh, yeah, we're moving on. This is not what I call big boy lab. There is no more lab quizzes because we're not doing like an IVDV here. There's nothing to graph. There's no measurable data. We're just doing this cool experiment.
And this is probably one of the most in-depth labs you've done, though, all year.
Yeah, totally.
So
you're going to take, and then we will go through these procedures so we can explain things. So you will take your graduate cylinder, measure out some water, and then put in about 20 drops of bromthymal blue. And then you'll pour that solution into the flask. The cool thing, the reason I want you to use a graduate cylinder and then put the drops in and then pour it, as you're pouring it, it also causes it to mix.
So you're going to repeat steps 1 and 2 so that by the end you have both flasks that have 100 milliliters of water that have the blue dye in it. So similar to the methylene blue, it is a dye that can stain. So please make sure you put the lid back on as soon as you are done. And get it off the counter.
Okay, so the cool thing about this this bromthymal blue is that it's also a pH indicator, which means that it indicates if something's acid or base or neutral. So you're going to actually take a straw, a clean straw, you're going to put one end into the flask, and you're gently going to blow into the solution, and then you're going to, as you're blowing into the straw, you're introducing carbon dioxide. When that carbon dioxide reacts with the water, it's going to create carbonic acid. Since it's creating an acid, it's going to change that blue solution. This is what you're going to start off with, blue. That acid is actually going to turn the solution to yellow.
Be careful when you're blowing with the straw. You want to kind of blow bubbles, but don't blow so hard that you're splashing it.
No splashing.
And also make sure, too, that you're paying attention. I know sometimes when you have a straw in your mouth, the tendency is to
go up.
Yeah, don't do that. You don't want to
down.
You don't want to drink what's in this flask. So.
No. If we see indications of horseplay, we just yank you from this lab, and you don't need to do this, which would be unfortunate because I believe there's references to this lab in your assessment.
Yep.
So I don't want to see any horseplaying. I don't want to see any tendency, because I've noticed some kids playing with droppers in the past, which is just stupid, to playing droppers with people.
Yeah.
This is a big kid lab, so we're treating you like big kids for letting you do this. Please be responsible.
So by the end of step 3, you should have 2 flasks that have both are no longer blue but have turned yellow.
Not the green. Keep on going. It does take it takes a while. Some kids are faster than others. Depends how much of hot air you have, I guess. Okay, so then you're going to go back to the Elydia, and then the last several times you've been taking a leaf. Now you need to take a section, and we don't need to take the whole stem. But
Just
a couple
like
inches.
That much, you think?
Right. But it needs to be equal. Whatever you grab from one, you need to grab from the other to control that variable. It's important to have just one thing you're changing. And you're going to put both of those into the flask. The reason why we're using flask and not beakers is beakers have a larger lip, and you can introduce the oxygen into them more. This is the narrow opening. Put the foil on top, and that prevents outside air from going into your flask. And then you're going to put your initials on it, and you're going to write dark or light. So the first one you'll label like JM, that's my initials. I'm in 4th hour, dark. And then on the other one, I'm going to write JM 4th hour light. So do your initials, label one your initials hour, and then light, and label the other one initials hour dark.
And the dark ones, we will have a place probably inside cabinets.
A cabinet. We'll label
that
it.
you can put, and we'll put a sign there. And then the ones that are bright, we'll either have a light out for you, or we will have you place them in the windowsill.
Yep.
Okay.
Okay. So again, you're going to want to write this chart down. You don't need enough. You can write the pH. This is kind of cool. Lower numbers, the lower the number, the more acidic you are. And the higher number, the more basic you are. But I would write this down into your notes. Yellow means it's an acid, which means you did introduce carbon dioxide in it, and it reacted with the water, creating the carbonic acid. And then the blue indicates that carbon dioxide has been probably turned back into oxygen. So if there carbon dioxide is present, it will be yellow. If oxygen is present, it will be blue. You need to consider this. If photosynthesis happens, which gas will be present?
Bum, bum, bum.
There is a lab sheet, so make sure, and we don't have it in front of us because we're not prepared. Make sure you're answering all the lab questions that go along with this before you get said and done.
Yeah, and this is a 2-part lab. So you're going to leave those flasks overnight, and then you're going to come back the next day and revisit them to see if there are any changes. And if there are changes, why might those changes have occurred?
And sometimes it helps if you leave them for 2 days.
Yeah.
In all honesty. Okay. When you do put your light one in the light, it helps to make sure you don't have any shadows. I've noticed in the past, if kids put foil over the whole flask, then of course there's no light going to be penetrating the flask, and no photosynthesis will happen. So make sure your foil stays concealed to the top of your flask, and you're not covering the whole thing. And then make sure you're also not putting it in a shadow, that it has direct light.
Right.
Any other thoughts?
Nope.
Okay. Kiddos, have a good day.
Later. Done.